Madam, start చేయండి.
ఏదో వచ్చింది స్క్రీన్ మీద విడ్జెట్ మెనూ అని వస్తుంది.
Madam, start the class.
Okay. Good morning, students. Yesterday we have learned up to chemical origin of life. Today we will start with biological origin of life. Actually, so this biological evolution includes the formation of protebionts, the origin of living organisms. Coming to the first point, that is formation of protebionts. Actually, till now we have learnt about the chemical origin. So during that chemical origin, organic molecules have been formed. So these complex organic molecules are called as conservates, and these ones will be synthesized due to intermolecular attractions. Actually, these are conservates are actually taken to be bubble-like droplets, and these ones will be seen in the primordial soup itself, according to Oparin. And these conservates, bubble-like droplets, will be attracting the substances which will be seen in the surrounding media due to intermolecular attractions. You know very well that each molecule will be having attraction towards the other molecule which will be residing beside it. Since these conservates are residing inside the primordial soup, they will be attracting other chemical molecules due to attraction between those ones. And apart from this, some type of chemical organizers called as conservates, termed to be free genes, will be attracted. Again, those ones will be forming all clubbed together, just like one organism type will be formed. And this one will be having the ability to take in molecules from its surroundings. And ultimately, surrounding this material, the lipid membrane will be formed. And this formation of lipid membrane will be making all those complex organic molecules to appear just like an organism, a primitive organism. That's why it is termed to be protobiont, pro in the sense first. So in the beginning, how the living organism was there, like that we have to come to the understanding. So what happens when this primordial soup or prebiotic soup has been formed in the ocean? The organic complex organic molecules will be absorbing the other substances from the surrounding prebiotic soup and will be forming a bulged structure, and surrounding that bulged structure, which is having free gene like chemical organizers, will be ultimately developing the lipid membrane. And due to the coverage of this lipid membrane, naturally it will appear as though it is an organism. So like that, this one will be formed. Now you can see this bacterial structure, how it will be. As you know, this bacterium is prokaryote. You know very well, in lower classes you might have learned prokaryotes are the first organisms which will be showing us all the living organisms characters. So this is the thing. And coming to the second point, the origin of actual living organisms. Here, the so-called free genes, that is the chemical organizers which have formed the protebionts, will be absorbing the organic molecules from prebiotic soup and have evolved into anaerobic heterotrophs. Anaerobic, you know the term anaerobic, in the sense without oxygen, they'll be going on living. And these ones are heterotrophs, in the sense they have to depend upon the others for their food material. So example, we can give Pseudomonas, a rod-shaped bacillus, that is nothing but bacterium. And here, these ones, in the sense, free genes with proteins uncovered with lipid membranes, having enzymes, all these ones will be starting the life as anaerobic heterotrophs. This is approximately 3 to 4 billion years ago. See how actually we are in the track of how these organisms will be getting evolved. In the sense, since the Earth's origin, how the life began and how they get evolved, all those ones we are on the track, is it not? So 3 to 4 billion years ago, this origin of anaerobic heterotrophs might have taken place. And these early living organisms had clumps of nucleoproteins containing 1 or 2 DNA molecules. As you know very well that nucleic acids are termed to be DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So if these nucleic acids are there, we can ultimately tell that it is an organism, living organism. So now we are in the path how these ones, in the sense anaerobic heterotrophs have been formed, and how these ones will be getting evolved into actual organisms. So for that, the DNA molecule should be there, and even now due to the addition of purines and pyrimidines, you can expect the DNA formation, and these ones are termed to be the present prokaryote. Beside this, you can see the prokaryote, a good example, that is nothing but bacterium, which will be having a single flagellum. This prokaryote, why it is called as prokaryote, you know very well. Pro in the sense first or beginning. In the beginning, actually how we are seeing it presents cells with nucleus enclosed by nuclear membrane. It will not be there like that in prokaryotes. Nuclear material will be there, but it is not enclosed by nuclear membrane. That is the actual difference between prokaryote and eukaryote. Eukaryotes will be having DNA, nuclear material, which will be enclosed by nuclear membrane. But in these prokaryotes, nuclear membrane will not be there. That's why we can take it as a primitive organism, primitive living being. Okay. So during this course of evolution, these prokaryotes have acquired the carbohydrate synthesis catalyzing enzymes. Previously itself, enzymes were there, and these prokaryotes, once they got originated, they started acquiring the carbohydrate synthesis catalyzing enzymes. And as a result, here chemoautotrophic organisms have been formed. These chemoautotrophic organisms can be differentiated into iron and sulfur bacteria. So, so many varieties of bacteria will be there, iron and sulfur bacteria. For example, if you take up the Gallionella and Leptothrix, these ones can be taken as examples for iron bacteria, and Spiribacter and Rhinhemera will be taken as examples for sulfur bacteria. So how these prokaryotes, a simple bacterium, acquired the enzymes that can synthesize the carbohydrate material will be ultimately taken as chemoautotropic organisms. Actually, these organisms, in the sense chemoautotropic organisms, were living in high temperatures. As we have seen in primordial soup, previously when the earth got originated, the temperature is actually 5,000 to 6,000 degrees centigrade. So in that hot temperature, these organisms are getting thrived, and these ones will be getting evolved into next step. So these organisms can take carbon dioxide from the atmosphere, and they can utilize that carbon dioxide for formation of carbohydrates primarily. In the sense, these ones are getting tracked into photosynthesis. You know very well that carbon dioxide in the air will be utilized by organisms, particularly our plants, and they will be synthesizing carbohydrates. Is it not? So here sugars will be primarily glucose formation will be occurring during the process of protein synthesis, photosynthesis. Like that, now primitive organisms such as bacteria have evolved into the organism such that they can utilize chemical energy. Okay, and among these organisms which were getting evolved, they can take sulfur bacteria example. So here they will be having special bacterial chlorophyll, which will be utilizing magnesium porphyrin. As we have seen, when the earth has been formed due to the hot temperature, rains occurred, then those ones got deposited into oceans, and all that one was called as prebiotic soup by appearing. Is it not? Can you recollect those ones? Yesterday I told you. So this, in this thing, since in oceanic water, magnesium is there, these primitive organisms have absorbed magnesium porphyrin, and they started trapping the solar energy and fixed the carbon dioxide. But unfortunately here, these ones, since they are utilizing H2S, they cannot give out oxygen. That means here carbon dioxide will be utilized, sunlight will be utilized, H2S will be utilized. Instead of water, hydrogen sulfide will be utilized. Due to this thing, of course, carbohydrate complex formation will be there, but we cannot expect oxygen to get evolved. Normally in photosynthetic reaction, you know very well that carbon dioxide plus water with the help of chlorophyll and then sunlight, it will be giving rise to sugar, that is nothing but glucose, and then oxygen will be released, water will be released, all that reaction you have learned in 10th class itself. But here, since these organisms, primitive organisms, nothing but sulfur bacteria, will be utilizing hydrogen sulfide, they'll be unable to release oxygen. Okay, so that is the middle stage now. We have learned here the origin of life, then the co-servates, then formation of primitive anaerobic heterotrophs, then prokaryotes, then now we have achieved this sulfur bacteria. Since these ones are utilizing the H2S, they are unable to produce oxygen. That means till this period, the atmosphere can be treated as reducing atmosphere only. You know very well, hydrogen is released now. Hydrogen is there in the atmosphere itself. So if hydrogen is there, naturally that atmosphere can be taken as reducing atmosphere. When we can call that atmosphere to be oxidizing atmosphere, if at all the atmosphere contains oxygen only. So we are in the process of evolution, how these ones got originated. Here, later, this bacterial chlorophyll evolved into true chlorophyll. So you know very well bacterial chlorophyll is there in purple-green sulfur bacteria, which have utilized H2S, but couldn't give out oxygen. This bacterial chlorophyll has evolved into normal bacterial chlorophyll, into true chlorophyll. This true chlorophyll can be seen primarily in cyanobacteria and plants. True chlorophyll in the sense, actually the chlorophyll which at present we are able to see in the plant body. So naturally this chlorophyll, true chlorophyll, will be able to absorb water vapor and release oxygen. So that capacity is there for true chlorophyll. As a result, here the large quantity of oxygen has been evolved or released, and ultimately reducing atmosphere has been changed into oxidizing atmosphere. So for this purpose, it took 1 billion years ago. 1 billion years time period was taken when the conservates have been formed, then anaerobic heterotrophs have been formed, prokaryotes have been formed, chemoautotropic organisms have been formed, then sulfur bacteria have been formed. All these changes, evolution is nothing but a change, is it not? So it's a continuous process, and it is a lengthy process and very, very slow process. Imagine how many years, in the sense billion years took just to get just to change the reducing atmosphere into oxidizing atmosphere. Since the oxygen is very much available, naturally you can expect all the organisms to get evolved one after the other, one by one, like that. Okay. Why we are able to exist now at present period, in the sense, since we are able to respire, intake of oxygen and outletting the carbon dioxide, is it not? For taking in oxygen, oxygen should be there in the atmosphere. So that oxygen has been evolved by these blue-green algae. That means the bacterial chlorophyll got evolved into true chlorophyll, such that it can absorb water, carbon dioxide, sunlight. With the help of this chlorophyll, naturally it will be able to produce oxygen. Like this, the reducing atmosphere has been changed into oxidizing atmosphere. Okay, and after this, these organisms are able to take up the oxygen in aerobic mode. Previously, we have seen anaerobic mode, is it not? Anaerobic heterotrophs we have seen 3 to 4 billion years ago. So these ones have been transformed or evolved into aerobic mode. Why? Because oxygen, free oxygen is available. Okay. So later these eukaryotes evolved further, probably by 2 processes. The first process is some prokaryotes entered the bodies of ancestral eukaryotes and lived symbiotically. Symbiosis, you know very well. So mutual help will be done there. And these prokaryotes have entered into eukaryotic body and lived symbiotically. And these ones have been transformed into mitochondria. You know very well about the mitochondria. Those ones are taken as the powerhouses of the cell, is it not? So how the oxygen that has been taken in to the cell will be giving out the energy just due to the processes that occur in mitochondria. In a sense here, aerobic bacteria have entered into ancient anaerobic prokaryote and transformed that anaerobic prokaryote into primitive aerobic eukaryote. How it is, it has been done just by the entry of aerobic bacteria and living symbiotically. So like that, they have been transformed into mitochondria. That means they help the organism to get evolved into aerobic eukaryote, and added to that, it further get evolved into phytoplanktons and plants in one side, and zooplanktons, fungi and animals on another side. Okay, so these ones here, you can see they evolved into organelles such as mitochondria and plastid. Plastid, you know very well, that is nothing but chloroplast. Chloroplast can be seen only in plants, and mitochondria can be seen everywhere. So no need of plastid for animals, zooplankton, all those ones. But for plant, phytoplanktons and plants, plastid should be there. In the sense here, photosynthetic bacteria have entered into this primitive aerobic eukaryote and got transferred into plants. Without their entry of photosynthetic bacteria itself, they have the primitive aerobic eukaryote has been transformed into zooplanktons and fungi animals. Actually, here the endomembrane system of eukaryotes might have evolved by the foldings of plasma membrane. You know very well, the cell membrane is there, that is nothing but plasma membrane. If it is getting folding inwardly, you can expect the formation of mitochondria, as we are able to see in the bacterium itself. Since the bacterium will not be having any other cell organelle, its membrane itself will be acting as the mitochondria. How it is possible? This plasma membrane will be infolded, and ultimately it will be acting as mitochondria, such that it can produce or release energy. Okay, so that's about the biological evolution. And now we'll go for the experimental verification of chemical origin of life. Here we have previously we have seen A.A. Oparin and J.V.S. Haldane. Those two scientists have proposed the chemical theory, is it not? In the previous lesson, previous class we have learned. So just to support this Oparin's theory, Urey and Miller, that is nothing but Harold Urey and Stanley Miller, have successfully proved the theory put forward by Oparin. Okay, so these two scientists have built or constructed one practical arrangement. There they have constituted the substances or environment which was there in previous periods when the life got existed or when the life get originated. So that circumstances, those arrangements were made practically in this experiment. For this, you know very well, they have taken soup, prebiotic soup. That is nothing but oceanic water. And they have boiled that oceanic water with the help of heat source. And see the aroma here, how it is going like this in the tubular structure, and have entered into one round bottle like structure, which will be already containing methane, ammonia, hydrogen, all these. Okay, you have to recollect the previous class, how these ones will be there in the atmosphere. Methane is there, ammonia is there, hydrogen is there. That's why we have termed that atmosphere as reducing atmosphere, is it not? So now this water got vapor and changed into water vapor, and all those ones got collected in this flask-like structure. In this to this flask, with the help of electrodes, the power was given, and ultimately so many reactions occurred in these chemicals. Okay, and later on they were pumped into downwards like this, and while they are traveling towards this down region, they were cooled. Hot water vapor joined with this methane, ammonia, hydrogen, all these ones got clubbed with the help of electrodes, and when they were traveling, that was cooled and got collected into this down region. So the collected material, when they have tested, they have seen, or they are able to see the material. See the material just like the simple amino acids. Amino acids are nothing but the building blocks of proteins. You know very well. So if these amino acids are there, naturally you can expect the formation of proteins. If proteins are there, naturally you can expect the organism to build its life or build its body. You know very well, our body is made up of proteins, muscles, all these ones. Here, the protein formation can occur just because of the union of amino acids. So the collected material in this region, when it is tested, they are able to find out simple amino acids such as glycine, alanine, aspartic acid, which are termed to be the non-essential amino acids. You know very well, amino acids can be diversified or taken into two varieties, particularly essential and non-essential. In previous classes, you might have learnt about this. There are 9 essential amino acids and 11 non-essential amino acids. Among these non-essential amino acids, glycine, alanine, aspartic acid, all these ones are there. So those amino acids, which are taken to be the building blocks of proteins, were the resultant of the material which we have collected in this region. Okay, so in this down region, they have collected and tested. So naturally, the formation of the non-essential amino acids you can expect. And later on, while they are doing further experiments like this, they have found out all the types of amino acids and also formation of adenine and other nitrogen bases also. Okay, so if adenine, in the sense, you know very well, it is part and parcel of purine, is it not? Purines and pyrimidines along with 5-carbon sugar will be forming nucleosides. Yesterday I told you. So when this nucleoside has been formed, if it is getting attached to phosphate, we can expect nucleotide. And if these two are there, naturally you can expect DNA, is it not? So DNA formation also can be expected. So these adenine and other nitrogen bases such as thymine, then guanine, cytosine, all these ones were seen as they further done the experiments. So like this, by utilizing the prebiotic soup, we can expect the origin of life. So this is the thing about the biological and chemical origin of life, nothing but post-surveyed theory put forward by, that is chemical is put forward by Oparin, Haldane, and experimentally proved by Urey and Miller. So this is about the thing. Now we will go for the actual evidences of biological evolution. Since we are dealing with the zoology topic, naturally we have to go for biological, is it not? Chemical we have to keep aside. What are all the proofs, how we can prove that biological evolution has taken place. Okay, so for this, many theories have been put forward. Of course, for anything, many theories will be there. Among those ones, we have to deal with the fact one, in the sense how, whether it is possible or not, whether it is true or not, we have to find out by experimentation method. Unfortunately, here no practical proof for that. Why? Because, as I told you, this evolution will be taking, it is a slow process, very, very slow, mild process. It will be taking millions, billions of years. Can you expect a human being to live during first year to million years? Can't, no? That's why we cannot observe even a single change in our lifespan. Hardly 100 years, 70 years, like that we can live. But we cannot live for billions and billions of years, is it not? So it is highly impossible for anybody, any scientist also, to observe the change that occurs in the organism's body. That's nothing but that favors the evolution. Hence, it is all hypothetical only. But even though it is hypothetical, we have to find out the evidences. What are all the evidences that will be showing us this hypothesis is correct or this hypothesis is not correct? Like that, we have to find out. For that, evidences should be there. Okay, and for these evidences, they have taken from different branches of biology. In 11th class itself, we have learned about branches of biology. So many branches are there: embryology, paleontology, anatomy, genetics, evolution, then the ecology. All these are treated to be the branches of biology. So from different branches of biology, they have taken hypothetical evidences, in the sense evidences from different types of branches of biology. So these scientists have taken evidences from paleontology, embryology, comparative anatomy, cellular and molecular biology. Like this, they have taken the proofs, in the sense evidences, and they have given us the knowledge of evolution, how it might have taken place. Might have, in the sense it is hypothetical. Okay, nobody can live and practically see and write the matter so and so like this happened. Nobody is able to do that. That's why it is only hypothesis. If hypothesis has been formed, we have to find out whether it is true or not. Whether it is true or not. Okay, for that we have taken evidences from paleontology. So now we'll go for the first one, branch of biology, that is nothing but paleontology. You know very well about paleontology. It is the study of prehistoric life through fossils, is it not? So by study of fossils only, we can come to know about the organisms' life which existed prior to us, before us. In a sense, here prehistoric life through fossils, it is treated as paleontology. Okay, and these fossils are remnants of plants and animals that were preserved in the layers of the earth, which have been excavated from the soil. So how we can find out the fossils? By excavating the soil layers, we can find out the imprints of or remnants of imprints of plants and animals. And by studying those ones, we can find out how they lived and what are all the structures that are seen in their bodies, and what is the type of food material they have consumed. So all these ones we can find out by observing or by studying these fossils. Okay, so these fossils have to be studied. And here there are various types of molds, casts, petrifications, coprolites. Coprolites are nothing but fossilized fecal matter. You just imagine fecal matter of the previous living organism, previous lived organism also can be seen. In the sense, by experimenting that fecal matter itself, we can find out the food material that has been consumed by that particular organism. Is it not? Just by observing or by finding out the materials that are been given out as feces. So like this, the study of fossils will be done, and actual remains of animals will be preserved in ice. Okay. And by studying these ones, we can have the idea of life that gradually evolved on this earth. As you know, here earth will be covered layer by layer by soil, and by excavating the soil layers, we can find out the fossils. And by studying those fossils only, we can find out the previous life. In the sense, downmost layers will be having ancient organisms. Topmost layers of the earth will be having just previous to us. So like that, you have to take gradations. In the sense, layers of earth, layers of soil. If you study downmost one, if you take up, you can find out the ancient, most ancient animals or plants or living organisms in general we can take. Okay. So how this one can be done in the sense by radiocarbon dating method. You know very well about this. Carbon 14 is utilized apart from uranium-238 and potassium-40. Generally, among these three chemicals, you can utilize carbon-14. Okay? So by utilizing this carbon-14 by radioactive radiocarbon dating method, you can find out the fossils life, in the sense when those organisms existed. So we have to go back into the periods, in the sense years, previous years. This is billions and millions of years. How we can go back means only due to utilization of carbon-14. That is nothing but radiocarbon dating method. Okay. So here the biologists and paleontologists have found the fossils of many transitional forms also. These transitional forms, in the sense connecting links. At present, we know very well about the classification, how these ones are there, then how phyla are arranged in animal kingdom, all these ones. You have studied some of the connecting links, transitional forms. So those ones will be seen in between the layers of the earth, such that we can find out that these previous organisms might have evolved into next organisms. In between these ones, connecting links also will be there. Okay, like that here, Eustenoptera can be treated as the connecting link between fishes and amphibians. Here, if you take up vertebrates, it is classified into 5 classes, as you know: Phaeaces, amphibia, reptilia, aves, and mammals. So in between these fishes and amphibians, this Eustenoptera is treated as a connecting link, and Simuria between amphibians and reptiles. Archaeopteryx, you know very well about this, since you have learnt in previous class. Archaeopteryx between reptiles and birds. Then Cynonatus between reptiles and mammals. Okay, so like this here, connecting links also can be taken as proof for paleontological evidence. Okay? So here this complete fossil records they will be preserving, and we can take up various stages. And till now we have we are having a very good history of horse evolution, how the horse got evolved. Previously, horse was not that much height. It's just like a dog height. But how it got evolved, what are all the stages that occurred in the evolution of a horse is available for us at present. Okay, so these are all taken into criteria how the evolution might have taken place. It's not a sudden change. Evolution is a gradual and slow process. Okay. So regarding this, just to find out this fossil records, as I told you, carbon dating method should be utilized. And regarding the study of these fossils, we should have a little knowledge about the geological time scale. In the sense, how the time has been divided, how we can go back to millions and billions and trillions of years back ago. If you see, if you want to go for those ones, there should be, in a sense, present time division, you know very well. Seconds are there, minutes are there, then hours, days, fortnights, months, years, like that we are dividing. No? Like that only, the previous period, previous time, rather to say, previous time is divided into 4 categories. Nothing but eon, era, period, epoch. What is the use of this division of geological time scale in the sense? We'll be very well able to find out in which time or during which time the particular organisms have evolved. The particular organism or the particular group of organisms might have flourished well or evolved into another varieties. All these ones we will be able to find out when we are able to know, or when we are having the knowledge of geological time scale. So previous time has been divided into eon, era, periods, epochs. For example, here 4 eons are there. In the sense, eons are nothing but the time that is from the beginning, in the sense from the origin of life on this earth. Origin of life on this earth. Hadean, Archean, Proterozoic, Phanerozoic. These are the 4 eons which are available for us. That means you have to imagine billions of years span between these each and every eon. Hadean, eon. After 1 billion years, you can expect Archean eon. And after 1 more billion years, you can expect Proterozoic eon. Then finally, Phanerozoic eon. Okay. So here, these eons have been divided into eras. There are 4 eras, like eons only. Precambrian era is there. Paleozoic era is there. Mesozoic era is there, and finally Cenozoic era is there. At present we are in Cenozoic era, and again this Cenozoic era is divided into different periods. In a sense, actually era is divided into periods. So many periods are there. Each era has been divided into many periods. And since we are living in Cenozoic era, you can find out the quaternary period and tertiary period. Primarily tertiary period will come, and now quaternary period. In the sense, each era is again divided into periods. Each period is again divided into epochs. Okay, see here, periods, eras, epochs. Eras is there, periods is there, and epochs are there. So if you want to see these years, time, we can currently, we human beings are in Phanerozoic eon, Cenozoic era, quaternary period, and Holocene epoch. Quaternary period is again divided into two epochs: Pleistocene epoch previously and Holocene epoch at present. Actually, this Holocene epoch began 11,700 years ago, it seems. So that is about the epoch which we have, we are existing now. And if you go for I hope you might have seen the movie, English movie Jurassic Park, where all these dinosaurs will be there, and hero family will be entering into that island where these dinosaurs are there. So they will be escaping from them. So all those you might have seen. So those dinosaurs which are belonging to reptilia evolved further, in the sense they got flourished well in Jurassic period. That means Mesozoic era is again divided into Triassic, Jurassic, and Cretaceous periods. So here, as we have seen, the vertebrates are having the Pisces, Amphibia, Reptilia, Aves, Mammalia. These 5 classes are there. So here, this as the evolution has taken place, these fishes flourished well. That flourishing period can be taken as golden period, is it not? We have learned in social studies, golden period of Guptas, like that. In the same way, during this time scale, golden age of fishes can be taken as the Devonian period of Paleozoic era. And if you take up the golden age of amphibians, it is Carboniferous period of Paleozoic era. And if you take up the dinosaurs, in a sense nothing but reptiles, it is Jurassic period of the dinosaurs. That's why the movie name is given as Jurassic Park. Okay, and here, if you go for the Cenozoic era, tertiary period, among that Oligocene epoch, origin of many mammals have been got originated. And at present, the quaternary period, Holocene epoch. Now we are living in that Holocene epoch. Great civilization of human beings has been taken. Now we can take this Holocene epoch, quaternary period, as golden age of human beings, is it not? So like that, we will be taking this thing here. This is regarding the time, geological time scale. So this provides most directive evidences for the concept of evolution. In the sense, as we study the previous time, we will be ultimately able to find out the organisms evolution. Okay, you can see this now. See, I told you already here, quaternary period, Cenozoic era. Of course, eons are not mentioned here. Eons will be here in this region. Era is there, period is there, epoch is there, and What is the significance of that particular time scale? Okay. So here this thing, epoch, if you see, Holocene epoch is there, and where civilization of human beings. Origin is different and civilization is different. You know very well, human beings got originated in Pleistocene epoch, but the civilization, now you are able to utilize your mobile phones and you are able to listen my class. That means civilization has taken place, is it not? That's why it is taken to be the Holocene epoch in Quaternary period, in Cenozoic era, and in Phanerozoic eon. Like that, we have to go back. Whenever any person asks you what epoch you are, in the sense, Holocene epoch we have to tell about. Okay, so this Holocene epoch began 11,700 years ago. That means flourishing of human beings has taken place during that period also. Further, it is getting evolved. How, you know very well, in my, in the sense, in my early stage, when I, in the sense, my schooling age, these computers were not there. But now computers are there, mobile phones are there. That means civilization has taken place, is it not? So like that, it is also taken to be the evolution. Okay. So how we are able to find out, how we are able to utilize all these ones we can tell. Okay. So that's about the evidences from paleontology. And if you go for the evidences of embryology, coming to the next topic, embryology is nothing but study of formation and early development of an organism. It is treated as embryology. Okay. So regarding this embryology, great scientist named as Ernst Haeckel, he is treated as father of embryology. Okay. Ernst Haeckel. And later on, von Baer is considered as father of modern embryology. Just simple embryology, in the sense, beginning of embryology is done is dealt with Ernst Haeckel. But later on, after him, embryology, modern embryology has taken place just by von Baer. He is treated as father of modern embryology. So if you observe the embryos of different organisms, actually here what Ernst Haeckel has done, in the sense, he has observed all the embryos of particular vertebrates such as fish, salamander, tortoise, chick, man. And early embryos of all these organisms look alike. As the time period gets on, the development takes place in embryos, and finally changes might have taken place, and they might have got changed into different organisms such as fish, salamander, tortoise, chick, man. You know very well how these organisms will be looking. In the sense, as they getting birth, born, how you can't expect the chick to be just like man, or you can't expect the tortoise to be like man. Like that, how, what are all the differences? That means as the time period gets on, the embryos will develop some special characters. Those ones are assigned for this particular organism. But previously, as and when the embryo has been formed, all the embryos look alike. That means here itself we can imagine evolution. Even though the organisms, embryos, primitive embryos are looking alike, they'll be getting evolved into different organisms which are completely different, not looking alike or not looking the same, as how the man will not look like chick, or chick will not look like fish or salamander, like that. How the differences might have taken place in the sense previously, in the beginning or in the starting itself, the embryos look all alike, but gradually changes might have taken place. Okay. So today, let us stop this, students. Okay. Thank you. Shall we end this class?